Dr. Doug Axe. We're responding to a number of good and thoughtful questions you received from our conversation about your book, Undeniable, about how there's not a naturalistic explanation for the origin of proteins, and they also bear the mark of intelligent design. We're going to take, if there's some live questions, type question in caps, and if it's on point and relevant, and it's succinct and makes sense, you said bring on the toughest questions. So
Sure.
Here's some that were sent in through X, through YouTube, commented on the video. This first one comes from a skeptic, and I'm just going to word it the way it was worded. For those dishonest Christians who claim the cell was designed, then one, how did God design it? Having designed it, then did he also make the first cell? If so, how? Now, there's a couple follow-up points to this, but the objection seems to be you can't claim that God did design it if we don't know how God designed it. Your response?
Well, it's very common for us to look at things that are made by humans and say, well, I know a human made that, but I don't know how. So that isn't a stumper to say, if you're claiming that God did this, then you have to be able to say how God did it. But another sort of thread that might be in the question is the distinction between the origin of something, the design of something, and the operation of something.
Okay.
And if you take your smartphone, it is a physical thing and it operates according to physical law, but it didn't originate by physical law. So those are distinct things. It's very possible for something it's very possible for us to be able to infer correctly that something was designed while not denying at all that it operates through ordinary physical law. That's typically the way human-made things operate.
Okay, both those points are really helpful. Part of me wants to add up there. If we're talking about God, then by nature, God has the means and mechanism to do this, just like God could speak something into existence from nothing because God is an all-knowing, all-powerful being. So the question about how God does this doesn't seem to me directly relevant or interesting, but you're also not saying that we can infer design in protein. Therefore, the Christian God and all the attributes that Christians believe in and would say we could know from Scripture and from some of the other arguments. Would you agree with that distinction?
You wouldn't be able to, you wouldn't be able to infer from proteins the attributes of
those
God.
attributes. Yeah. Okay. Totally fair distinction. Okay, good.
Can I say one other thing on that?
Please.
Because I think putting myself in the position of the person who asked the question, if you think about the world in naturalistic, materialistic terms, you do think in terms of mechanism and mechanism only because that's all there is, okay? So a Darwinist, materialist, naturalist, and a lot of my friends are I'm not, there's no pejorative in here. If you think of the world in those terms, then you think the most important question about the origin of something is the detailed mechanism because that's all you have is a mechanism. But if you put yourself in the shoes of a theist, that no longer is the most important question. If God created something, then I don't need to know the sequence of atomic events. I know that God is adequate to do it, and it could have unfolded in lots of different ways. In other words, a material sequence is not of paramount importance to a theistic understanding, although it is to an atheistic or a materialist understanding.
That's a really good and helpful distinction. Of course, when we're looking back in history, we still want to know, even if we believe there's evidence of intelligent design, when did this first emerge? Why did this first emerge?
Yeah.
Did it happen once? Did it happen multiple times? These are scientific questions that are super interesting. You're just saying we can't minimize all of these explanations down to mechanistic explanations. When we see evidence of intelligent design, that's the heart of your argument.
Right.
All right, so this comes from somebody on Campus Crusade for Christ staff. Affection there because my parents are on Cru staff and I grew up as a Cru kid. So I'm going to go to this one second. We have some more skeptical questions coming up as well. This one says, Dr. Axe, your research seems to show that a random mutation search would likely fail to produce even one functional protein fold in the history of life on Earth. Does this fact also have implications for the plausibility of common descent / macroevolution in general, since building new animals would require the creation of many new protein and protein folds, not just one?
Yes, absolutely. So if you look at the huge variety of living things on Earth, you can see, even if you don't do biology, you can see how different they are, and they solve different problems. A butterfly has to solve certain kinds of problems. A bacterial cell has to solve certain kind of problems. An amoeba and we solve certain kind of problems. And by that, I mean biological problems, how we get our food, how we get our energy, how we live our life. So if you do drill in and look at the molecular level, many of those different problems require different suites of proteins, enzymes, all kinds of molecular apparatus to enable creatures, organisms, to do what they do. So this explosion, this huge variety of living things, and everyone agrees on that. There's a huge variety. That's not controversial. And it's also not controversial that this explosion of huge variety of living things does depend on a huge variety of different molecular components, proteins, in order to catalyze different reactions, in order to build different molecular machines, et cetera. So the person who posed the question is exactly right. If a blind mechanism cannot stumble upon a single functional protein, then it would have a very difficult time stumbling upon the many that would be needed to build a new life form.
Good. Yep. You're the one who really challenged me. We had a conversation about a year ago with my friend Sygart, who evolutionary creationist. You argued that the origin of life itself requires massive amounts of information, but the following steps through common descent up to us today is a greater leap than life itself. That surprised me. And some of the challenges you raised, Sygart went back and looked at him and said, you know what? I think Doug was right about this. The literature does not justify this in a way he had been led to believe, and people often claim. So we won't sidetrack in that conversation, but people can go back and watch it. It was a wonderful exchange between two believing Christians who differ over some of the mechanisms at play here. And I thought great came out of it. All right, friends, we're here live with Dr. Doug Axe, Caltech trained, Berkeley trained, colleague of mine here at Biola University, teaches biology. Here's the skeptical question. I know you've thought of this one, so we're going to go back to the live one. It says, is this hypothetical designer itself complex? So this is similar to the Richard Dawkins objections he made in the God Delusion years ago, about 20 years ago now. If the designer is complex, what have we explained by invoking it? If the designer is simple, aren't you just doing the same thing that you've accused naturalists of doing, namely asserting that complexity came from simplicity without providing any details or plausible mechanism?
I don't want to weigh in on the theology of the simplicity of God. I've heard people argue. I'm not a theologian, so
totally fair.
But let me say this: that any worldview, when it grapples with origins, has to grapple with a first cause. Okay, so whether you're an atheist or a agnostic or a theist, you have to believe that there's something from which everything else arose. You can always trace things back. And if you're a materialist, you might say, well, the Big Bang is where the universe arose. Well, how did the Big Bang arise? And you always trace back to something that is the first cause. So what a theist can claim, I think, rightly, is that God, if he exists, is a very plausible first cause.
Great
An
answer.
almighty, all-knowing, all-powerful God, if he exists, is a very, very good first cause. Now, and I know an atheist would say, well, the problem is he doesn't exist. But to that atheist, I would say, can you provide from an atheistic point of view a comparably plausible first cause? And I don't think you can.
That's a great distinction. And of course, God as by definition, an uncaused, self-existent, eternal being, in some ways akin to the laws of mathematics that are not the kind of things that can be caused, or like laws of logic, although personal, are candidates for self-existent, uncaused things.
Yeah.
When we look at proteins, there's a certain design element that you argue that we need an adequate cause for, because it's not in the same category as what a God would be in.
Right.
And so what is that explanation for the origin of this specified complexity? Cries out for a cause in a way that God, by definition, doesn't.
Correct.
Totally fair. Good response. William Lane Craig has written a lot of responses to this. You may accept it or not, but he's one who's waiting carefully as a philosopher. Okay, good response. Okay, how about this one? Here's a skeptical one. I like to come back to this. It says, Biola requires a statement of faith that you sign in order to attend the university. That is a problem before we even get outside the box. You already are starting this research from a biased point, which is going to taint the purity of intellectual honesty of any experiment or work coming out. That is a huge problem. Looking at the science and where it goes, period, without religion is more honest type of work. So it does not matter what this guy, presumably you, says.
That's me.
He went on your show and would not be on your show without the statement of faith mandate. Now, just for the record, before you weigh in, I've had a whole lot of people on my show, regular, including over the past couple of weeks, of people who cannot and will not sign a Biola mandate. So I have people on regularly who see things differently. But it seems the objection to you is that your research can be disqualified because you've signed a statement of faith and begin with the conclusion in mind and are tainted rather than following the evidence where it leads.
Yeah, there's good and bad in that. I mean, I agree with aspects of that strongly, namely that regardless of what our biases may be, science is one of these enterprises, and reason and logic are these enterprises that we can agree to disagree on some things, and let's put things on the table that we can agree on. And when the question says that follow the evidence where it leads or something to that effect, I totally agree. That if you and I disagree on our worldview, we can still do science together, and we can adjudicate some things by doing science together and reasoning well together. So I think that's a good point. Where I think the question is off the mark a bit is it seems to imply that Christians are unique in having a bias, and I would say that every human being has a worldview. It is the framework by which you make sense out of reality as you live your life. Everyone has that. Some are more reflective and self-conscious about the fact that they have a worldview, and some are less. So if someone says, I don't have a worldview, you do, you just haven't learned about what a worldview is. Everyone has it. It's a framework by which you make sense of reality. The second thing that I think is true of all people is they have a bias in favor of their own worldview, because we all go through life with a grid for interpreting things, and it's always a little bit painful to think, oh my goodness, the way I've interpreted everything my whole life is wrong. I have to change this. So you always, in the first instance, are trying to fit things into the grid that you have. So it's not proper, or it's confused to say we don't need to listen to that guy because he has a worldview. Well, you're not going to listen to anyone if you rule people out for having a worldview. What you have to do is the second thing that the person said is, okay, I disagree with that guy's worldview, and I know that he's got a bias in favor of his worldview. Yeah, but so do you, and so does everyone else. So we all have this confirmation bias when it comes to our own worldview. The way we adjudicate is exactly what the questioner said. Put the science on the table. Now, I've done that. And when the person says, out the gate, we don't need to listen to this guy because he's a Christian, now you're not actually doing what you said. What you should do, if you believe what you said, is look at the science that people are producing, even if they are Christian, even if they have a worldview that differs radically from your worldview, and let's adjudicate this on the science. And I claim there that the design argument has beat the Darwinian argument, and I'm saying this in public, and I put out public challenges. If anyone thinks that they can, and didn't I articulate this in the last time we spoke about the difference between the origin of stars and how astrophysicists can lay out 1, 2, 3, 4. These are things we know to be true, and they add up to an explanation for the origin for stellar origins. If someone can produce something like that, things that we know to be true that add up to the blind watchmaker thesis, which is that all of life descends from a common ancestor and a material cause is adequate to explain that descent, then I will change my view. I am so confident they can't, and no one's even come forward with an attempt. The thing about this kind of challenge is you have to put your money where your mouth is. If you think you can do this, start putting your statements down, and I guarantee you will either put down a statement that you claim is true that is not verified, and there's no way to verify it, or maybe it's been refuted, even worse, or you're going to put down a bunch of statements that are true, but they don't add up to this big claim. So that's my challenge.
I actually think signing a doctoral statement is beneficial in the sense that it makes very clear what our biases are.
Sure.
It makes very clear what our operating worldview is. And we're doing science that intersects with people who have other worldviews. And it's not like we're operating in separate silos. You have your worldview, they have yours. We make predictions, like you just said, and then we can assess the data. So yes, there's a real strong danger that if my job is at stake with changing my position, it builds in incentive to not believe something that's going to cost me a job. I have a colleague here who held certain views about the end times that did not match up with Biola's, and he stuck with his convictions, was not determined by them. So we've got biases, we're aware of them, but we can intersect with people with different biases and then see where the evidence leads. So it's a concern to watch, but it doesn't control the outcome, and there's nothing dishonest about it when our biases are made very clear.
In my case, let me also say, I came to Biola late career, okay? So look at the other places I've been, UC Berkeley, Caltech, Cambridge University, Department of Chemistry, Medical Research Council Center in Cambridge. None of those made me sign a faith statement, but I was a Christian at all of those. And did I have a bias as being Christian? Sure, we all have a bias. So key aspects of my work have come out, not since I've been here, to be consistent. If someone is complaining about the fact that I've signed a faith statement, that didn't change anything. I came here because this place allows me to do the work from the position that I held at other places that don't make you sign a faith statement.
So we're here with Dr. Doug Axe. I would consider one of the leading proponents and supporters of intelligent design, author of the book Undeniable. And I mentioned that because one quick question that came through is, what are the best books to read on intelligent design?
Well, you just named one.
That's exactly why I started with that. Now, of course, when we say the best books, are you talking about kids, academics? Like, you have to qualify all of these things. I wrote a book with Dembski in 2008, and it's still somewhat evergreen because there wasn't an introductory text to intelligent design called Understanding Intelligent Design. We're updating that right now, which is fun. Undeniable, I would definitely recommend. Any other books jump to mind for people on intelligent design? Just one or two that you think would be helpful.
Well, Michael Behe's classic is Darwin's Black Box, 1996-ish.
That's right.
Steve Meyer, my colleague, has written three books. One of them is the basis of a film coming out very soon, The Story of Everything. But his book is called Return of the God Hypothesis. So that's another good one. I really like his first one, which is Signature in the Cell.
It's a great book.
So there's good books on different things, and they focus on different things. I think there might be, and I've had a conversation about what is there like for a middle schooler. There might be a need. I know there are some things for a younger audience, but I think there's a need for more there, to be honest.
The closest would probably be a Case for Creator from Lee Strobel for kids.
Is there a kids version?
For teens. There's a kid version.
Okay.
Now, one of the differences is Lee Strobel is explicitly and clearly a Christian evangelist writing for that audience. Intelligent design per se doesn't identify who the designer is. And that's why you'll have people from different faith commitments embrace intelligent design. That is not remotely a critique. I agree 100% with Lee Strobel. But there's a difference between just saying, here's intelligent design and what the science can point towards, and making a case for Christianity with the intelligent design arguments. But for students, that is the best one I'm aware of. I would start there. Case for Creator for kids, Case for Creator for, for teens or for students. Okay, let's go. Here's another skeptical one I find interesting, shorter. It says, it's more of a statement than a specific question. Somehow apologists want to say there's no conflict between faith and science, and then turn around and say, there's a massive anti-faith conspiracy among nearly every scientific institution and professional in the world. So this person is pointing out at least what they think is a conflict between Christians who say there's no conflict between faith and science, they can be reconciled. And then the number of Christians, namely ID proponents, who say there's a massive anti-faith conspiracy in these scientific organizations and professional organizations and universities in the world. Is this a fair point about a contradiction among apologists?
Yes, I think it's conflating two things, both of which are true. When people of faith say we don't believe there's a fundamental conflict between faith and science, I'll call it capital S science, the most pure pursuit of the truth by observing the natural world. And using reason, the human intellect, to do honest observation and honest discussion about how best to explain things. If that's capital S science, then that's not going to disagree with faith, not with Christian faith, because they're both true and they both come from the same creator. But science as practiced can look very different from that, and very different from capital S science. In fact, it's interesting that the person who wrote the question used the term scientistic, not scientific.
Yeah, that was interesting.
Because scientism is really a religion. It's the idea that science is the only path to truth. And there's no way to show that to be true through science. So it's something that people hold to be true basically by a faith position. So yes, scientific institutions are at odds with the Christian faith because they have a different faith that's contrary to the Christian faith. So it's not as though one is pure and intellectual and the other is tainted. Scientism is tainted, and you can't do science well if you think science is the base reality. You can't consistently do science if you think science is the only way to arrive at truth, because the question, how do we know science is valid, cannot be arrived at by doing science.
And there's other things within the scientific pursuit, like morality, that I'm going to report the truth on my findings for future generations, which is assumed within the scientific pursuit, but cannot be established and known scientifically, to further your point. I would argue, we don't have time to go into this, but when apologists argue there's no conflict between faith and science, I think Alvin Plantinga is right, where he argues where the conflict really lies is between naturalism and between science, not faith and science. I think some of the conflict is on the surface, age of the earth, certain understandings of evolution. But when you get down to it, there's a reason the scientific revolution took place in a culture shaped by a Judeo-Christian worldview.
For sure, yeah.
I would argue the conflict, there is no conflict there. And it's secondary whether or not organizations are affected by scientism and persecuting Christians. That's a separate question that has nothing to do with whether science and faith itself has a conflict. All right, good stuff. We're making some progress here. Here's a sophisticated one. Oh, man, some questions came in. Let me see if I can find this one. Okay, so some of these I sent you ahead of time. This one says, shared genetic markers like endogenous retrovirus insertions and the fusion evidence in human chromosome 2 seem to strongly suggest common ancestry. How would ID explain or predict these?
I'm presuming common ancestry with the great apes is what I'm reading into that, because
doesn't say, but that's fair.
I think that's what we're saying here. So if we do find genetic properties, genetic features in humans and in chimps, we'll say, there's different ways to explain that. One way would be that we humans share a common ancestor with the chimps, and something happened, something broke, and the chimps have their modern version of the thing that broke a long time ago, and humans have their modern version of the thing that broke a long time ago. And I think if we knew that something is truly a random, if you could look at the genome and identify something and know for sure that it is a chance random thing that's very unlikely to have happened this way and not a billion other ways, that's a strong argument. But I think it's very much harder than most people claim to know that. So there are things, even things that are attributed to retroviruses, we don't know exactly what's going on in the genome. We don't understand these genomes that well. And so the reason that is relevant is if there are aspects of the genome that look like something is just broken, but actually it isn't a broken gene, it's a switchable gene, it's something that is under some control of some kind, we may be mistaking a designed feature that actually does something for a broken random accident.
Which we've done many times in the past.
And time again, yeah.
Going back to vestigial structures, et cetera.
Right. So in that case, if you're actually looking at something that is this way for a reason, for a functional reason in humans and in chimps, well, we could start naming all kinds of things. We both have two eyes. We both have a spine. There's all kinds of things that are that way for a reason, a functional reason, that we have in common with chimps and dogs and guinea pigs and rats. And there's a reason for that. My point being, it's the critique of design has often very quickly jumped to the idea that this is junk DNA. And junk DNA, as a hypothesis, has been largely discredited in the decades since that term was first coined. One would need to know that one is looking at something that's truly junk, has no functional purpose whatsoever in order to make that argument stick.
That's an assumption that was built into, if this is cobbled together through a blind materialistic process, a lot would be junk. Ideas, including people like Jonathan Wells, predicted that we could test and find purposes for DNA, and we've actually found many distinct, important purposes for DNA. And by the way, for clarification, we were asked earlier, what are the best intelligent design books to read? You mentioned Darwin's Black Box by Michael Behe, who believes in common descent.
Yeah.
Now, I don't know the arguments he gives for common descent, but that is not an essential primary question intelligent design wrestles with, nor does it wrestle with the age of the earth. It doesn't start with the Bible. It's whether or not living systems bear the marks of intelligent design, which point towards a mind as the best cause. So whether or not there actually is common descent is secondary and arguably somewhat irrelevant to the case ID is making. Not irrelevant to the science, but irrelevant to the case ID is making. Is that fair, or would you nuance or critique that in any way?
Yes, absolutely. And there are many different views. I don't agree with Mike on that. I don't think there's compelling evidence for universal common descent. I think that's what the question is referring to. Are all living things related in a tree of life? Mike Behe, I don't think he argues that, but I think he holds to it, and I think he holds to that loosely.
Yeah, he holds to it, but still sees things like the flagellum and blood clotting is like, however the processes came about is secondary.
That's the key thing. So one question is there a tree of life? Darwin's tree of life, is that a real thing or not? One could say, yes, I think that is a real tree, but you don't get any branches on that tree without God intervening and designing the things along those branches. And that would be roughly Michael Behe's position. I question the tree itself, but yes.
Got it. So do I. Fair enough. Okay. Excellent response. Okay, this one's a little bit longer, but I think important. And by the way, we're here with Dr. Doug Axe, a colleague of mine, friend, one of the leading defenders of intelligent design, author of an excellent book, Undeniable. This is our Talbot Tuesday. Although you don't speak at Talbot, this intersects with what we do, or you don't teach at Talbot. You speak for us all the time. Intersects with what we're doing here. And we're responding to the video you did, making a case from proteins to an intelligent designer. If you have live questions, I've been trying to take as many as I can right here, write question in caps and state it distinctly, clearly relevant to the topic. We'll take it. Back to one posted earlier, Doug. It says, the teleological argument states that the probability for life developing in the observable universe are astronomically low. Therefore, an intelligent designer is so much more plausible than chance. Atheists respond by saying, if the conditions of the universe were different, thereby prohibiting life as we know it in the universe, then a different kind of intelligent life, one that we don't know and have no concept of, would have evolved and came into existence. So they say the fine-tuning is just meaningless and no designer is actually needed because different conditions would bring different life. Now, this is actually, more that I read it, this is not an objection to your work. This is more of an objection to fine-tuning, which is in physics and cosmology. But I wonder how you might respond to this objection.
Yeah, two things come to mind. First of all, that's kind of a gaps argument that whatever universe you get, I claim life can evolve, and I don't have to show you anything. That's a very, very weak. It's okay for scientists to have hypotheses or start with hunches that are that weak, but you wouldn't roll that out as a serious scientific theory to say whatever the parameters were, something could produce intelligent life. You'd have to show something before most people would take that seriously. That's point one. Point two is, let's just frame the argument with this universe, because we know this one exists. What the intelligent design community is saying, in a universe with properties like this one, you will not get life apart from intelligent causation.
Good response. Now, I want people to know I'm actually super pumped about this. It's Friday, posted an in-depth interview with Jay Richards, friend of yours, friend of mine. He's also at Discovery. He was also here in studio on the fine-tuning argument and the privileged planet. I specifically asked him this question. And the point he makes was, it's not that if we didn't have fine-tuning, we'd have different chemistry and different life. We wouldn't have planets and stars and even the possibility of any kind of life whatsoever. We're going to get into that post it Friday, and then he will join us next Tuesday, 4:30 live. Jay will be here taking your toughest questions on the fine-tuning argument.
In fact, relevant to the film that's coming out, The Story of Everything.
Yes, take
One
a
of the
minute.
things I learned from the film, I didn't realize how incredible the prediction of Sir Fred Hoyle was in the film, where he realized that for stars to be the element factories, and so that's the way we believe cosmology works, that you start off with a universe that's hydrogen collapses under gravitational compaction to form stars through fusion, and the stars are making helium. Stars go through their life cycle and explode, and you end up getting heavier and heavier elements produced as you go through star cycles like this. Well, Hoyle realized that there's a pathway to get carbon-12, which is crucial for life as we know it. And he realized that there is no way that this would work unless there was a very specific resonant energy in carbon-12. He did the calculations and said, for these two elements, I think it's helium and beryllium, to come together and give you, I might be wrong in that. I'm recalling it's helium and beryllium. Cut me some slack. For them to come together and give you carbon-12, you would have to have a resonant frequency of the nucleus of carbon-12 at this particular frequency. And he said, if that's not there, then there's no way stars could give you carbon-12. And so he goes to these scientists at Caltech and said, look for this. And they thought he was crazy. But one of the guys looked for it, and there it is. Bing! Right on the money. A resonant frequency that allows carbon-12 to be formed by stars. So I only learned of that when I watched the film. That's an incredible validation of the fact that this is a universe that has exactly those properties, seemingly intentionally, that it would need to have in order for elements to be produced, in order for life to work.
So go see the movie, The Story of Everything, that's out. It's incredible. I saw an early release of it and was blown away. B, Jay Richards shares that story in depth on the video that we're posting. And one of the arguments that he makes is that fine-tuning was not discovered by Christian apologists trying to argue for God.
Yeah.
Fred Hoyle was an atheist, maybe an agnostic, discovered this surprisingly, and then we found dozens of other examples since. And as Jay argues on Friday, there is no good defeater for fine-tuning. You think I'm blowing smoke? Watch it Friday, join us Tuesday, post your questions. Jay will take your toughest questions. We will not ignore any. So let me bring you back to another tough question that came in your land. I've got two in a row that seem to come from people who understand the arguments, and these are sophisticated. Dr. X, you said the number of organisms to ever live on the planet is somewhere around 10 to the 40th. Is this referring to individual organisms or individual species? Does this include all biological life, such as bacteria, viruses, fungi, plants, animals, including both land and marine? You refer to organisms specifically, not species, so what I assume but I just want a clarification on whether it's an individual or species and if it includes all forms of life. Now, that one was less skeptical. The next one will be so, but it's asking for a clarification on the number of organisms to ever live on the planet. Is that what you meant by 10 to the 40th?
Yes, I would not include viruses. Viruses are particles that can hijack a living thing, but they're not themselves a living thing. And I don't think that's controversial among biologists. The number wouldn't be that different if you did include viruses, but 10 to the 40th is a reasonable estimate for the total number of living individuals of any species of any kind that have existed in the history of life on Earth. If I Googled that recently, and something like, some people estimate like 10 to the 30th alive today.
Okay.
That might be a little bit on the high end, but if you take 10 to the 30th alive today and you say in a year, there might be 1,000 of those, 1,000 times that, because it's mostly bacteria. We don't go through 1,000 cycles in a year, but bacteria do. You'd have 10 to the 33. If you say in a billion years, you add another 9 to that, you have 10 to the 42. So somewhere, it's somewhere in that ballpark.
That's fair. There's estimates worked into the models is the point. Okay, this one says, a genuine and respectful question. In your
I like those.
I like the way it's phrased. In your 2004 paper, weren't you testing mutational tolerance in a deliberately engineered, temperature-sensitive, low-function beta-lactamase variant rather than a normal, robust enzyme? If that's right, why should the rarity estimates from that fragile starting point be taken as evidence that functional proteins in general are isolated in sequence space or beyond the reach of mutation and selection, especially since similar functions can arise from neighboring sequence families and even different protein folds?
Yeah, there's been a lot of work went into that paper, and a lot of work went into that starting point. And it has been misunderstood since then, mostly by people who are outside the field. It went through peer review, so I can tell you, it went to the top-notch journal, top-notch peer review. Janet Thornton, head of the European Bioinformatics Institute, was the editor in charge, sent it to 3 top-tier reviewers. They didn't see a problem with that. So let me explain, but I understand why, if you're not in the field, it might sound like, hang on, he crippled the enzyme and then he found out how tightly constrained the sequence is. Well, the short answer is, do you want to find out the sequence constraints on a highly perfected enzyme or on a very loosely, wobbly, barely working enzyme? If you want to find a large target, it's the loosely held together, barely working, wobbly enzyme, not the perfected one, right? Because if you're asking how could evolution produce something, I conceded in the paper that the target evolution would have to hit is the larger target. It's the barely held together thing that's barely working. That's the one I set out to measure. If I had set out to measure the target for a highly refined, it would have been a far worse number.
Oh,
than I got. Far worse.
Far worse for the naturalistic assumption, not for your case.
It would be a much tighter, a much smaller probability to hit a high level of function than to hit a low level of function. So a lot of thought went into how do you get a starting point that's relevant to the origins question, and a highly perfected starting point is not so relevant to the origins question.
Okay. I think
did I land that bird? I know it is a little bit complicated, but
I think so. Fair enough, though. Yeah. Good stuff. Okay, I'm looking through the comments here. Let me go back. There's one I might take, but let's go back to some that were posted before. I think I've taken all of these right here. Okay. How directly can you apply the research you did on your protein of choice to other proteins? Most especially what a materialist might claim are early functional proteins. How short would an amino acid sequence need to be in order to reasonably arise from chance, in your opinion? So maybe, let me come back and read this again. Briefly, to an outsider, remind us of what your case was for proteins, and then let's come back and I'll follow up with this question of how much it would apply towards others. So just give us a quick recap.
Of how I got the result.
Yep.
I took an enzyme called beta-lactamase. It's the enzyme that allows bacteria to survive in the presence of penicillin, because penicillin binds in the active site and it breaks the penicillin. So that's the starting enzyme I looked at. I took one, it happens to have kind of two halves to it. There is one protein chain, but a lot of chains fold in multiple domains, they're called. So I looked at one domain, the larger of the two domains, and I messed with it. I left the other half exactly the way it was, and I thought the best way to measure how finicky this thing is to mess it up and see what fraction of the messed up versions work. Now, I was only asking it to deliver a very low level of function. And there again, that's why I brought it down to a low level function. And then I said, I just want to know how hard is it for you to maintain, to acquire a low level of function. So I had it at a low level of function, then I messed it up and I said, how many of these messed up variants still have the low level function? If I'd done this with the high level of function, it would have been a far more scary. It would have been 1 in 10 to the 120th or something. It would have been a much worse number than 1 in 10 to the 77th. So it was done by multiple experiments where I messed up a cluster of amino acid side chains, produced hundreds of thousands or millions of variant genes, put them into bacterial cells, and then put the bacterial cells onto Petri dishes with a very, very low amount of penicillin. But a high enough amount of penicillin that if it doesn't work at all, it won't form a colony. So it still has to work, but it doesn't have to work robustly the way the wild type one would work. And when you do that sort of experiment, you can then literally count colonies on this plate, colonies on that plate. I can count the fraction that works, get numbers, multiply out. If the whole thing were randomized in that way, what would you expect? And that's how you get the final number.
Okay, got it. Very helpful. So simply as possible, you're taking an existing protein, kind of breaking it, so to speak, and have a low threshold of what functionality would be, trying to test exactly what.
So you start with this robust, extremely good version of an enzyme.
Yep.
You weaken it by putting in multiple changes that make it less stable, less functional, but it still provides more function than you can get without an enzyme. Okay, so this thing can allow bacteria to survive in the presence of, I think it was something like 10 milligrams per liter penicillin, which would kill normal bacteria, but the robust one would allow you to put a gram, like 100 times, 200 times more penicillin, and it would still destroy the penicillin. So you start with that weakly functional, but functional, significant functional starting point, and then you do shuffling in that to see, well, how hard is it to hold something together this well, okay? And we've shown in other experiments, you can take that weakly functional starting point that we did, and you can take it through rounds of mutation and selection and build a very, very highly, you can optimize it, but you need an actual folded enzyme in order to do that optimizing.
Okay, so I think that relates directly to this question. Arguably, is it one thing to break something down and check its functionality versus the origin of that thing itself?
Yeah.
So they're saying, how directly can you apply this research you did on your protein of choice to other proteins, most especially what a materialist might claim are early functional proteins? So what relevance does this have for the origin of proteins and other proteins?
Well, as I said, if I had done the experiment differently and taken a very, very exquisitely optimized enzyme and measured the sequence specificity for that level of function, try saying that 10 times fast, I would have got a much more scary number. So I realized that people are going to want to interpret this in terms of a constraint on protein origins. And to get a protein to originate, you just have to get it to fold, maybe loosely, not highly stable, and it has to have the right things in place to do its job. And then, as I've shown, as I and my colleagues have shown, if you have one of those weak starting points, you can evolve in a Darwinian way a very highly functional protein, but that's a much more constrained thing. So the relevant question is, how hard is it to get a protein sequence that does the job even poorly, but it does it by folding and having an active site? That's the relevant question. That's the question I believe I gave an answer to in that JMB paper. The questioner may also be asking, okay, you did this on one protein.
Sure.
beta-lactamase, how relevant is that to the thousands of other proteins? And there I did argue briefly in the paper. I actually compared it to another protein where people did something like this, but it was a much smaller charismate mutase. And they're in the same ballpark. The numbers are in the same ballpark.
Oh, interesting.
Yeah. So I argue, although if you look at these folded structures, they're very different, they still are built almost exclusively of alpha helices. It's the corkscrew-shaped part of a protein, and beta strands, which is kind of like a zigzag shape of a protein. Beta strands form beta sheets. These are the two secondary structures, they're called, that Linus Pauling predicted before they were confirmed. He got the Nobel Prize for it. Smart
Amazing,
guy.
Yeah.
And because you build all protein structures basically out of the same kinds of things that have to come together and stick together, and they use the same kind of physical forces to stick together, I argued that by sampling the various regions within one protein, you're really sampling a general problem, which is how hard is it to get a protein chain to collapse and be stably folded. And obviously, if you sampled that on more and more proteins, more data would be better, but I argued that this gives us a reasonable number for solving that general problem.
Love it. Here's a good skeptical question, I presume. It says, if you accept microevolution, dog breeding, etc., but not macroevolution, could you explain at what point natural selection stops working and why? Now, I would say Behe's more recent book, Darwin Devolves, is exactly devoted to this question. And it talks about one mutation, but when you start to get to two multiple coordinated mutations to build something requires foresight, and the blind mechanism can't generate that. So in some ways, an answer to this, Behe's book, Darwin Evolves, gives the depth and the details as somebody wants to. But where would you say, because you accept microevolution, but not macroevolution, at what point does natural selection stop working, and why do you think it stopped working there?
Yeah. So an interesting thing about Darwin Devolves, he gives the polar bear as kind of a... The polar bear of Darwin Devolves is the mousetrap of Darwin's black box. It's his
That's right.
showcase example. And in the polar bear, Behe argues, I think quite persuasively, that brown bear, so grizzly, is slightly more, has the full genetic complement. It's a genetically, very obviously, polar bear is genetically complex too. But he argues that polar bear evolved from brown bear by genetic loss, not by genetic gain. So there are genes that get broken in order for this big bear to specialize in different niches. And that's what he argues polar bear is. Now, that's an interesting idea. And if it's more general than just polar bear, it could be an explanation for how you get specified specialist versions of different animals and plants that do very well in particular niche environments that have come from a more generalist version of the organism, and they've come from there by genetic loss, not by genetic gain. That's a very interesting hypothesis.
Okay.
So what that means is evolution works when it doesn't have to come up with new stuff. Evolution works when it can break existing stuff. So that would be the Behe answer. I would say more generally, that's the problem, is evolution is not good at inventing. It's very good at sorting. So classically, natural selection sorts things that exist, and if something that does the job exists, then you're going to get that. That's what selection will give you more of the thing that already exists that works. What it won't do is figure out a new way to solve a problem to make a new thing work.
In other words, if you go from, what do you say, a brown bear or a grizzly bear to a polar bear, it's still kind of a horizontal change.
Yeah.
Creating new vertically, that's where it gets beyond the realm of natural selection to do that. And you would presumably include not just natural selection, but evolution today. There are other mechanisms that contribute to this, not natural selection. The question was just on natural selection, but you would say that is a larger problem regardless of the mechanisms that are added to natural selection as well.
Yes.
Fair enough? Or
yes. What's missing is an adequate explanation for the origination, the origin of complex new features that solve new problems. No one has solved that. Random mutation, I think most people know is not adequate, but you can trot out a whole bunch of other ideas. None of them, they look good on paper and you can get books written about them, but they never actually deliver the goods. That's the thing that's missing. Not the, what's the famous expression was, natural selection may
explain the survival of the fittest,
but not the arrival of the fittest. So that's the theory that's needed.
That's great. Okay. I think I'm getting a good amount of the objections here. I'm not trying to ignore any ones that look tough. Let me come back. This is a great question I know you've thought about, kind of a personal question from somebody who accepts the ideas you're putting forward. It says, for a young person interested in ID, what should I study at the university to explore new frontiers in ID? Where does the case for intelligent design need improvement?
That's a great question. There's been a sense among kind of the old timers in modern ID, and I'm one of them, that it's been my sense too. I spent a lot of time critiquing Darwinism because I felt like, okay, here's the thing that we need to actually demonstrate doesn't work. And not just my work, but lots of people have shown it really doesn't work. But after you've done that for decades, I feel like I reached a point where I felt like we are beating a dead horse here. And okay, there are still Darwinists out there, but they just need to wake up and smell the coffee, so to speak. The evidence is all out there to show that this doesn't work. More interesting to me and to a number of others is, okay, suppose everyone came out and said we're post-Darwin now, and all the research universities said, okay, we've abandoned that idea. How would we do what new frontiers in biology would open up?
Good question.
Because we view life from a designed perspective. And I have some ideas and some colleagues have some ideas that they're pursuing, but I think there is, and really the last chapter in my book, Undeniable, talks about this, like a road to a new version of biology. I think there's all kinds of things that would become new frontiers if all the people who are spending an awful lot of money doing biology research had this lens from which to do the research. And I think we're only scratching the surface. There are meetings where we get together and say, we should be looking at this, we should be looking at this. But I think if it were 1 million life scientists as opposed to, we might be able to scratch together 1,000 now, it would look very different. And if they were well-funded, it could look very different.
It seems to me that if we go back to 90s and early 2000s, when ID was really exploding onto the scene publicly, even though some of the work had been done by like Charles Thaxton or Walter Bradley going back to, say, the 80s, fine-tuning going back to, I don't know, decades possibly before then. But then there's like these novel ideas that come out. You have the privileged planet thesis. You have your arguments in protein. You have William Dembski's arguments. You have irreducible complexity. And Lee Strobel's book, Case for a Creator, really captured kind of this moment where they came together. I haven't seen any in like the 20 years that came out, these new novel kinds of arguments that seem to explode on the scene in that moment. Is moving forward now saying, let's look for novel arguments like we did then, or let's keep defending these, let's advance these, let's fine-tune these arguments, or should it be both moving forward?
I've been involved in one new thing, and it came out of a Evangelical Philosophical Society meeting in, was it 2024, November 2023, or it's probably 2023.
Okay.
No, it's 2024. I can find out. But Bill Dembski and I were in San Diego in this session that was on design. And I gave a new probabilistic argument that I thought, here's a very simple probabilistic argument that I think is a takedown of accidental cause of living things or any complex things. Dembski was there scratching notes down. And then, as he does, a month or two later, he sent me this manuscript that he said, what you said in that conference gave me the clincher to finish his proof of the conservation of information.
Wow.
So he now has a proven, this is now published, conservation of information theorem, and he drew on just a little example that I gave at
That's
this
awesome.
talk. Yeah. So I think that's very cool. And I've now articulated this. So I have a chapter in a volume that's coming out soon, I hope, where I've articulated my take on this as well.
Is this a volume on ID or a volume on...
It is a volume that covers multiple perspectives.
Okay.
And I think I won't give details on it until I know it's...
I got some reluctance. I understand.
So that's a new thing that I was involved in. And I do think it is a proof in a way that people on my side of the argument were thinking, there's got to be a way to nail this as a proof and not just a bunch of arguments. And
nice.
This is fresh.
I love that.
But I also think, it's probably yes to multiple things. We have to keep training up people so they know the standard arguments well, because I've already had colleagues who've been involved in this passing away, and there's a realization that we're not here forever. And so we want good arguments to be robust and have longevity, and that comes by training up people in using the arguments, but also this other project of, okay, forget about arguing about something that in some ways is a dead horse. I think it's also interesting that the most accomplished people are not, you don't see them on the stage arguing against design. When Jim Tour went up against Professor Dave, that's a highly accomplished scientist, and there isn't a highly accomplished scientist opposing him. It's Professor Dave, who's not actually a professor, opposing him. And that's interesting, because where are the Robert Jastrows and the people that you see in this film, top-notch, top of their game astronomers and cosmologists who are coming to a realization, gulp, it looks like the atheistic view of this doesn't work. And there's several examples in the film. I'm also thinking that very, very highly competent life scientists are aware that there is a major failing in the naturalistic account of life, and they're being kind of quiet about it right now.
Dembski and I are working on an update to the book, Understanding Intelligent Design. So I will be moving back into some of the arguments in a way I did almost about 18 years ago. So I am eager to kind of get back into the weeds on this and kind of learn from a generalist how the argument has shifted. But I think your idea of advancing new arguments, and I want to read that paper, and fine-tuning the ones that exist makes sense moving forward. All right, folks, pick up a copy of Doug Axe's book, Undeniable. It's sophisticated but very readable, and you've defended it ably here. One last question I think is a good one to end on. This comes from Mark Corbett. Shout out to Mark. He watches comments on a lot of videos. Appreciate you. It says, I'm not sure, but it feels like opposition to ID is more intense and harsh in the biological sciences than it is in physics. Do you sense that this is true? If so, why might this be?
Yes, I do sense that. And I think if you see this film, The Story of Everything, you see a bunch of astrophysicists who are, grappling with something they're not really, they're being forced to acknowledge something that they didn't want to acknowledge. And I think you see that in Fred Hoyle, Robert Jastrow, Dean Kenyon, a number of these scientists. But they can do it. And they got up, there's clips, Alan Sandage, getting up there in front and saying, look, I don't see why we need to shrink back from the fact that this demands a theological explanation. And these are people who are not theists coming to that conclusion.
Yeah.
If you come to that conclusion as a life scientist, I think to the general community, it feels as though you are pulling the plug on kind of a foundational theory for understanding what life is, and that's Darwin's theory. And after a conference, in a closed door, small group setting, you can get scientists, biologists who are not ID friendly to acknowledge that the thing's a mess. But they won't do it at the podium. They won't do it in publication. They won't do it at the podium. And so I think there's a sense that there's a danger to what will happen to the life sciences if everyone acknowledges that the materialistic understanding of life has failed, materialistic understanding of life's origins has failed, and everyone's very afraid of, what would happen if we open the door to intelligent design. I think actually good things would happen, but I understand.
I agree, 100%.
I understand why people are nervous.
I think there's a difference between the origin of the universe, the fine-tuning of the universe, which still feels a little distant and abstract, although I think we get certain attributes of God arguably from this, and protein in the biological world and in the human body having a designer. That gets a lot more personal.
It does.
And that's where I think a piece of this is at play, the worldview behind it. You've mentioned this movie, The Story of Everything. Again, I've seen it. Fantastic. We're live streaming this on April 21st. I realize some of you will see this later, but tomorrow at Biola, 7 p.m., we're having a screening of this on campus. So Wednesday, April 27th, a screening on campus. Stephen Meyer will be here to answer questions.
April 22nd.
Didn't I say April 22nd?
You said 27th.
22nd, let me clarify. Definitely April 22nd.
Yeah.
You'll be here. Stephen Meyer will be here answering questions. You'll get to watch the film early before its release in theaters.
On the 30th. That's the big release.
On the 30th. Good. These dates are important. Now, Friday, if you're like, I'm not in Southern California, I can't get there. I'm doing a deep dive with Jay Richards on fine-tuning, walking through what's the strongest way to make this argument, what are the best objections against it, how has this argument shifted over the decades. We had a lot of fun. He sat here, he came in studio, even though he lives on the East Coast. So I'm going to post that on Friday. And then a week from today, April 28th, we're going to do the same thing you and I did, where I'm going to gather the toughest questions that people can send in. We will have Jay Richards with us, and then we'll take some live questions as well. This is a part of our Talbot Tuesdays, something that's been new since March. Comment, give us a thumbs up. Let us know. My team will be reading this. Do you want us to continue this for some time? Pick it up back in the fall. Is this a helpful service, whether you're a skeptic and you're a seeker, whether you're a student, to do these kind of live conversations with Biola Talbot professors and others? I think I covered it all. Did I miss anything, Doug?
No, you got it. You nailed it.
This is fun. We will